Okay. Good afternoon. I'm Carolyn Smith. I'm the Interim Senior Vice President and Dean of Research, and it's my pleasure this afternoon to welcome you to the Michael E. DeBakey Award for Research Excellence. Each year, we are pleased to recognize exceptional members of the Baylor College of Medicine faculty for their contributions to clinical and basic science. These awardees are representative of the quality and scope of research at Baylor, and their published scientific contributions over the last 3 years have demonstrated significant achievements and contributions to their field of study. And in so doing, their efforts have contributing to improvements in human health and patient care. These awards are named in honor of the pioneering heart surgeon Michael E. DeBakey, the first president of Baylor College of Medicine, and they are sponsored by the DeBakey Medical Foundation. I'd like to thank the DeBakey Medical Foundation for their continued support of these awards and welcome the Medical Foundation President, David Short, who I understand is joining us via our video link, and our trustee, Bill McCollum, who is joining us here today. Thank you for being with us. The DeBakey Medical Foundation was first established in 1961, and through Dr. DeBakey's groundbreaking advancements in cardiovascular surgery, he impacted the lives of countless patients and established this organization to continue the momentum of innovation and advancement in healthcare. Before we get to the awards and presentations, we have a special message today from our Executive Dean and President, Dr. Paul Klotman, who could not be with us today. Do we have that? We don't have it? Claire is not here today. Okay. Hopefully we can track that down before we get to the end of our presentation. Sorry about that. So then, to begin, we are honored today to recognize 6 outstanding researchers here who are members of the college. And our first award today goes to Dr. Robert Atmar, who's the professor of medicine in the section of infectious diseases and holds appointments in molecular virology and microbiology. As a leader of the National Vaccine Treatment and Evaluation Unit Network, Dr. Atmar's work played a critical role in the national research response to the COVID-19 pandemic by significantly advancing our understanding of SARS-CoV-2 vaccination responses and helping to define strategies for boosting immunity to both the original SARS-CoV-2 strain and emerging variants that remain in clinical practice today. With the impending approval of several COVID-19 vaccines in the fall of 2020, Dr. Atmar proposed the first adaptive clinical trial to evaluate heterologous vaccination strategies, anticipating correctly that boosters would be needed. His work demonstrated that boosting with a heterologous COVID-19 vaccine was safe and led to antibody responses that were at least as good as those induced by homologous vaccine boosting. The data was critical for the FDA's and the CDC's decisions to make heterologous COVID-19 boosting available to the public. And the resulting publication in the Journal of the New England Journal of Medicine has since been cited more than 400 times as of January of this year. It was recognized by the Infectious Disease Clinical Research Consortium as the network's scientific paper of 2023 and received a 2023 Physician Enterprise Vision Awards Academic Excellence in Clinical Research recognition from Baylor's clinical partner, Catholic Health Initiatives, which is now Common Spirit Health. His studies also demonstrated the ability of boosters to induce heterologous antibody responses to emerging COVID-19 variants, contributing to the adoption of U.S. clinical practice guidelines that recommend inclusion of mRNA vaccine in a patient's initial COVID-19 vaccination series. In addition, his evaluation of Novavax as a heterologous booster demonstrated that it had limited benefit to those with hybrid immunity and could safely be used by persons who do not take the mRNA vaccine, presenting a strategy that is logistically simpler for healthcare systems and patients and introduces additional competition into the COVID-19 vaccine market, saving countless resources and lives. So with that, I'd like to present you your award. Leave it to the side, okay. Congratulations.
Thank you.
Okay. Oh, and this is yours. Oh, yeah, and then we sit down, we're gonna present all the awards, and then we will have our talks. Our next awardee is Dr. Christine Beaton, who's a professor of integrative physiology and academic director of the Cytometry and Cell Sorting Core. Dr. Beaton is a world-renowned investigator in the treatment of autoimmune diseases and recognized for groundbreaking research focused on the identification of novel therapeutic targets and treatments for rheumatoid arthritis, and new, highly effective, non-invasive drug delivery systems. Her work was amongst the first to identify potassium channels as druggable targets on effector memory T lymphocytes and fibroblast-like synoviocytes in rheumatoid arthritis, leading to the development of recombinant analogs of sea anemone and scorpion venom toxins that block potassium channels on effector memory T cells with high affinity and selectivity. She's also demonstrated efficacy in animal models of rheumatoid arthritis and other autoimmune diseases. Of these peptide analogs, in their work on them is a subject of an investigational new drug filing for phase 2A clinical trials. And the company running those trials has assigned an option to license the patents on other analogs. A huge hurdle in the delivery of small peptides as drugs for chronic diseases is the need for repeated injections. In studies published in PNAS in 2023, Dr. Beaton and her colleagues bioengineered a probiotic bacterium to secrete one of her NV1.3 blocking venom peptide analogs for oral delivery. Feeding rats with a bioengineered probiotic was shown to be effective in delivering detectable levels of the analog into circulation, and the orally delivered formulation was more effective than injectable peptide in stopping disease progression in a rat model of rheumatoid arthritis. Targeting potassium channels with very selective and potent modulators provides a completely new approach to the treatment of rheumatoid arthritis without immunosuppression. The oral delivery of the peptide KV1.3 blockers via the engineered probiotics has the potential to revolutionize peptide drug delivery, offering a non-invasive approach in avoiding injections. This approach is now being tested on other peptide potassium channel modulators, and international collaborations are underway to apply this delivery technology to other peptide therapeutics. And I'd like to welcome Dr. Beaton up and to receive her award. Thank you. Oh, Aaron. Okay. Thank you. Congratulations. Oh. Our next award is for Dr. Jeffrey McGee, who is a professor of neuroscience, and unfortunately, Dr. McGee cannot be with us here today. Dr. McGee is a world-renowned neuroscientist whose work focuses on the hippocampus, a critical brain area associated with spatial navigation, learning, and memory. Since coming to Baylor in 2017 as a Howard Hughes Memorial Institute investigator, Dr. McGee's work has answered several long-standing questions regarding neuroscience and how environmental information is stored and updated in the hippocampus. He first described a novel neuronal plasticity mechanism, behavioral timescale synaptic plasticity, which has revolutionized our understanding of how behavioral experiences convert into permanent synaptic hinges in the hippocampus. Additional work provided compelling evidence of BTSP's role in updating the hippocampal map based on experience, demonstrating its necessity for learning modifications in hippocampal population activity. This research also showed that entorhinal cortex layer 3 neurons generate an instructive signal to drive BTSP representations, which is a connection critical for normal learning and memory. A separate study described the dynamics of place cell presentation and how they evolve over time and adapt to new information, demonstrating that different populations of place cells have different levels of stability. Importantly, the activity of behavioral timescale synaptic plasticity has shown to shift the population to more stable representations, providing further evidence for the role of BTSB in storing information about the environment into an expanding long-term memory representation that can be readily retrieved. Together, these studies have important implications for both basic and clinical research in essential brain mechanisms, and particularly the development of novel therapeutic strategies to improve memory function in diseases such as Alzheimer's that feature disruption of hippocampal function. So I'd like to thank and congratulate Dr. McGee, and we'll make sure that he gets his award when he is back to campus. Our next presentation is to Dr. Maxim Mamonkin, and Dr. Mamonkin is an associate professor of pathology and immunology and a member of the Center for Cell and Gene Therapy. Dr. Mamonkin's work focuses on developing engineered T-cell therapies for hematologic malignancies and alloimmune pathologies. His pioneering approach to eliminate malignant T-cells using CAR T-cells to target an antigen present on both normal and malignant T-cells has been groundbreaking in the field of translational immunology. With BCM collaborators, he has developed a first-in-humans phase 1 clinical trial which showed that the CD5-CAR T-cells are safe and can induce clinical responses in patients with relapsed and refractory CD5-expressing T-cell lymphoma. This approach has licensed to March Biosciences, who have received CPRIT funding for a multi-center phase 2 clinical trial to develop this therapy for commercial use. Another major focus of Dr. Mamonkin's research addresses the need for cell therapies to transition to use of banked allogeneic cells. To overcome rejection of such products by the recipient immune system, Dr. Mamonkin has armed T cells with alloimmune defense receptors that recognize the expression of T cell activation markers, and has translated this strategy into the clinic. This work was licensed to Fate Therapeutics for use in inducible pluripotent stem cells, and Allogene has used these edited T cells, illustrating its broad specificity. In addition, Dr. Mamonkin has made significant advances in targeting malignant T cells to create new therapies for alloimmune pathologies driven by aberrantly activated T cells. His work has led to the development of a new class of receptors called alloimmune defense receptors that effectively eliminate activated T cells without targeting normal circulating T cells in their resting state. For these major conceptual and clinical advances in cellular immunotherapy, Dr. Mamonkin has been recognized by the American Society of Gene and Cell Therapy with an award from them in 2023 for an outstanding new investigator. He's also been recognized by the Texas Academy of Medicine, Engineering, and Science with their 2023 Prestige Award. With that, I'd like to congratulate Dr. Mamonkin and invite him up to receive his award. Our next award recipient is Dr. Leonid Metzelitsa, who is a professor of pediatrics in the Division of Hematology and Oncology. Dr. Metzelitsa is nationally and internationally recognized leader in the field of natural killer T cells, or NKT cell research, whose work has had a transformative impact on the development of pediatric cancer immunotherapy, including the rapid transition of basic research discoveries into innovative cancer immunotherapy clinical trials. Dr. Metzelitsa is the director of the Center for the Advanced Innate Cell Therapy and leads research in the development and clinical testing of cancer immunotherapy at the Texas Children's Cancer and Hematology Center. He and his colleagues have spent nearly 20 years developing innovative GMP-compliant methods for manufacturing genetically modified NKT cell products at clinical scale. This work has resulted in the first-in-human clinical evaluation of CAR NKT cells in children with neuroblastoma and represents a major milestone in cancer immunotherapy. The encouraging results from this trial support the premise that NKT cells can be safely infused in patients and mediate objective responses in children with relapsed or resistant neuroblastoma. Importantly, this paves the way for the development of next-generation NKT cell-based therapeutic approaches that can be used for the treatment of cancer and other diseases. Other major achievements include demonstrating that NKT cells mediate anti-tumor activity by attacking tumor-associated macrophages, revealing that tumor necrosis factor alpha triggers inflammation that promotes neuroblastoma growth, and identifying the LF1 transcription factor as a key driver of central memory program that drives NKT cells and their anti-tumor activity. So I'd like to welcome Dr. Metzelitsa above and congratulate him on this award. And finally, I'm pleased to recognize Dr. Bing Zhang as our final Michael E. DeBakey Excellence in Research awardee. Dr. Zhang is a professor in the Department of Molecular and Human Genetics and is a trailblazer in the field of proteomics and proteogenomics. Dr. Zhang's work has left an enduring imprint on global initiatives and scientific strategies aimed at combating cancer by significantly advancing our understanding of cancer, improved data analysis methodologies, and broadened data accessibility for the scientific community. His work has spearheaded data analysis endeavors and plays a leading role in providing comprehensive molecular characterization of multiple cancer types. Dr. Zhang's research is broadly relevant, as exemplified by his inclusion on the highly cited researchers list from Clairvaux. This includes 2 papers published in Cell, the first of which established a link between endothelial cell remodeling and immune exclusion in pancreatic cancer, and identified potential therapeutic targets with implications for early detection or intervention. The second paper pinpointed potential therapeutic vulnerabilities in lung squamous cell carcinoma, including the identification of retinoblastoma phosphorylation as a marker for trials using CDK4 and 6 inhibitors and other targetable points of immunoregulation. Dr. Zhang has made notable contributions in the field of computational proteomics through publications addressing crucial gaps in proteomic and phosphoproteomic data analysis methodologies, work which merited him the prestigious Gilbert S. Almond Computational Proteomics Award in 2023. In a highly cited 2023 Nature Communications publication, Dr. Zhang's team introduced a novel method to enhance the characterization of protein isoforms, a long-standing challenge in shotgun proteomics. thereby rendering the new tool highly valuable for a wider range of biological and translational research endeavors. Dr. Zhang's work also focused on enhancing accessibility to large and complex proteomic and multi-omic data for the broader scientific research community. In his role as the co-chair of the NCI's Clinical Proteomic Tumor Analysis Consortium, a pre-cancer resource working group, Dr. Zhang collaborated with other colleagues to harmonize a proteogenomic data set encompassing over 1,000 tumors across 10 distinct cancer types, and in so doing, created a unified and powerful data set that facilitates scientific discovery. To democratize the raw proteomics data and make them available to a wider audience, Dr. Zhang's team has established a web portal which enables users to directly query raw proteomics data and to quickly identify proteomic evidence for any known or novel peptides of interest, such as putative neoantigens. This work was published in Nature Communications in 2023. And I'd like to welcome Dr. Zhang up to receive his award. So I'd like to congratulate all of our distinguished honorees, and now we'll kick off the symposium portion of this afternoon's program. Any chance we've gotten the talk worked out for Dr. Klotman? No takers, okay. All right, hopefully we'll have that before the end, or I'm getting a message. We'll play it at the end, or can you play it now?
No, it was in an email.
Okay, we'll play it at the end. Okay, so sorry about that. So we are going to start off then with Dr. Robert Atmar, who will give us his presentation today.
Thank you, Dr. Smith, esteemed colleagues and guests. It's a pleasure to be here to tell you a little bit about the work we conducted during what was a somewhat stressful time for most of us. I'm going to take you back to the spring of 2021, and at that point, 3 COVID-19 vaccines had been approved for primary immunization and use in preventing COVID. But in that spring, the question came up, are we going to need a booster? And, we found out since then that yes, we would. But at that time, we were asking, when would we need it? What should we use? Who should we give it to? And would it be safe? We knew that pharmaceutical companies were pursuing homologous boosts, but from a public health standpoint, heterologous boost, in other words, vaccinating with a different vaccine than what one would originally receive, would be important. And so we designed our study, which is an adaptive design, which basically meant we could change it as we went along based on the information we got back. We enrolled individuals, adults who had received an EUA-approved COVID-19 vaccine at least 12 weeks earlier, so at least 3 months earlier. And the reason we picked 3 months, which was somewhat early, was so that we'd have the information in time for the fall to help guide vaccine recommendations that were anticipated to be needed at that time. We enrolled 50 participants per group based on the primary vaccine they had gotten and the booster they received, and divided that roughly up into people 18 to 55 years of age or 56 and older. And we didn't really design it to test any hypotheses. The information we were collecting was to guide public health decisions. This is a multi-center trial done at 10 different clinical sites. around the country that are associated with our Infectious Disease Clinical Research Consortium and have vaccine treatment evaluation units. Dr. El Sali leads our vaccine treatment evaluation unit here. So the initial part of the study was to evaluate over 3 stages boosting with 3 different vaccines. The Moderna vaccine at a dose of 100 micrograms per booster. This was the dose that was being used for prime. You may know that subsequently booster doses were approved or authorized at a 50 microgram dose boost, but we didn't have that information at the time we designed the study. The Janssen or the adenovirus vaccine 26 was stage 2 and Pfizer was stage 3. And then we enrolled people who'd received priming with one of those 3 different vaccines. So 3 by 3, 9 groups initially. And the primary objectives were to evaluate safety and reactogenicity and to evaluate the breadth of the humoral response. So this slide just shows that these vaccines were somewhat reactogenic. On the left part of the graph are the systemic reactions. On the right part are the local reactions. Basically, it made people feel bad for a day or 2 and gave them a sore arm, which was much what was seen with the priming vaccines. And most of the symptoms were mild or moderate, which are shown in the lighter or mid gray. The black were severe reactions that were reported. In terms of immune responses, this shows serum neutralizing antibodies as measured with a pseudovirus neutralization assay at Duke. And what we have are the 3 different boosters, the mRNA 1273, which is the Moderna vaccine, the Janssen Adeno 26, CoV-2, and then the Pfizer BNT 162b2. And then under each of those groups are the 3 priming regimens, Adeno 26, the Moderna, and the Pfizer vaccines. And on the bottom, you can see geometric mean fold rises, which is basically just a change in the level of antibody from pre-vaccination to post-boost, along with what the geometric mean titers were, expressed a couple of different ways. And to summarize all of this, basically people who got vaccinated had higher levels of neutralizing antibody after vaccination, which is what we thought would happen and what we hoped to find. And those who received a heterologous boost, in other words, a different vaccine than what they had originally received, had at least as high antibody boost as if they had received a vaccine with the same regimen that they originally received. The other piece of information here was that the adenovirus 26 vaccine didn't perform quite as well as the mRNA vaccines, at least in terms of looking at neutralizing antibody responses, which went along with the emerging vaccine effectiveness studies that were out. So these data were published in the New England Journal, but before that publication, you can basically see what the timeline was. We launched the study in the spring of 2021. Over the summer, we heard about myocarditis in young or in adolescents, particularly adolescent males who received an mRNA vaccine. Pfizer vaccine got licensed, and then in the fall, we presented the data to the FDA vaccine and related biologics advisory group and then it was authorized by the FDA. We presented the data to the ACIP work group. The ACIP recommended that heterologous boosts could be given similar to homologous without any difference in recommendation in terms of one versus the other. And the FDA then authorized booster vaccines, both homologous and heterologous boosts. We continue to conduct the study and enrolled 3 more stages looking at a bivalent vaccine, the mRNA 1273-211, which contained the beta variant and the original strain, and then the standard dose of the Moderna vaccine at 50 micrograms, and then a Novavax vaccine. the next question we asked was, okay, we measured immune responses to the ancestral strain. How about immune responses to some of these emerging variants? And the starred groups are the ones that we looked at in particular. And on this slide, you can see in the light blue for each of the different vaccine groups. So just to orient you, on top of each panel is the EUA vaccine that they received. So for example, in panel A, they received the Moderna vaccine at 100 micrograms, and they got boosted with the Moderna vaccine at 100 micrograms. And then the titer is on the y-axis, and the ancestral strain is shown in the blue dashed line with the BA.1 Omicron variant. You go back and remember the emergence of Omicron in the orange. And what you can see for each of the booster regimens is that the antibody response was greater to the ancestral strain, which was the immunizing strain, than to the Omicron variant. But we saw antibody responses to the Omicron variant in all of these booster regimens, and the expectation was that they would protect, and in fact, subsequently it was shown that they did. Here are some additional levels. So the further we looked at the emergence of Omicron variants from BA.1 to BA.4/5, the lower, relatively speaking, the antibody response was compared to the ancestral variant, but we still were able to measure responses to even the most distant at the time, the BA.4/5 variant. And then finally, we looked at the Novavax vaccine. This was a different vaccine based on expression of a protein in a virusome and used for immunization. There were a lot of people who were holding out for the traditional protein-based vaccines. But one of the questions that hadn't been asked was whether this could be used as a heterologous boost. And they were late to the game in terms of getting their vaccine authorized for primary immunization, and its primary use then would be as a booster. And once again, we did not see any safety issues, and looking at a variety of different variants, starting with D614G or ancestral strain on the top in the light blue, all the way to the bottom, the XBB.1 Omicron variant at the time, we saw antibody responses regardless of what people were primed with. So from this study, we're able to demonstrate that heterologous boosts didn't have any safety concerns based on the relatively small numbers we looked at, and antibody responses were similar to or improved relative to the homologous boosts. This information was used to inform public health vaccine policies and recommendations. It set the groundwork for a follow-up multi-center study of bivalent vaccines that were also conducted through our network. And then in data I wasn't able to show you today, we also established that boosting with an mRNA vaccine could lead to mucosal antibody responses that really set the threshold of the bottom floor for what would be expected for a next generation COVID vaccine response to try and prevent not just illness, but potentially infection. And it takes a lot of people to do these studies, many of whom are listed here. I want to highlight in particular my co-PI, Dr. Kirsten Leik, who was at the University of Maryland, and then Dr. El Saleh, who led our VTU here.
So thank you very much for that excellent presentation. If you have questions for Dr. Atmar and our other speakers, please bring those to the reception that we'll have after our event this afternoon. And with that, I'd like to invite Dr. Christine Beaton up to come give her presentation.
Thank you. So I'm going to be telling you about using a venom toxin as a drug, but also how we can deliver it better to make it better for the patients. So why are we using venom toxins and what diseases are we targeting? So we're targeting autoimmune diseases. And you all probably know at least one person who has an autoimmune disease. It's a disease in which your immune system attacks your own body. You may know someone with multiple sclerosis, rheumatoid arthritis, or another of those autoimmune diseases. And in those diseases, currently the treatments are mainly immunosuppressants. And so the patients can be immunocompromised. So obviously, if you're trying to make vaccines, then those patients may not be responding to vaccines as much as you would like. We know that the major population of T lymphocytes in those diseases are effector memory T lymphocytes. And we've shown years ago that those T cells express a potassium channel that is expressed only at very low levels on other immune cells, making it an interesting target for targeting especially effector memory T cells. Now, when you're trying to make a selective blocker of a potassium channel, you can go to chemistry, and you can make complicated chemistry, but you're getting either high potency or selectivity. It's very difficult to get both. But if you go to nature, and you look at all the small venomous critters, like scorpions, spiders, sea anemones, they all contain very highly potent toxins in their venoms. And a lot of those toxins target ion channels. So this sea anemone here that's shown on the left, Sticodactyla helentus, that lives in the Caribbean, makes SHK, which is a very highly potent blocker of these Kv1.3 channels. But it wasn't selective. So we partnered with peptide chemists, modified it, and we got SHK186, now of clinical name dalazatide, that is not only extremely potent on Kv1.3, but also extremely selective on Kv1.3 over other ion channels. So we tested this compound in vitro and in vivo in animal models. And then we did a phase 1 clinical trials. And I'm highlighting here the phase 1b clinical trials. So this was a double-blind, placebo-controlled trial. It was not powered very high, right? Those trials are made to show safety of the compounds and not efficacy. But we chose a disease, which is psoriasis. It's an autoimmune disease of the skin. And we chose a population of patients who had resisted to every existing treatment. So those patients were not taking any medication, nothing had worked for them, and those patients were not going into remission and spontaneous remissions. They got dalazatide injected 3 times a week in the abdominal fat pad, and they got 30 micrograms per patient. So we're talking minute amounts. This is not a typo, this is really 30 micrograms per person. And then we did imaging and skin biopsies before the start of treatment, shown on the left. You see the knee has a very visible skin lesion, and a month after treatment, you can see the lesion is dramatically reduced. Same with the hand. This patient could not move the hand. The skin would break and bleed. At the end of treatment, this patient could move the hand, had regained function. So that was very encouraging, and we are now moving dalazatide into phase 2 clinical trials. But there remains issues with dalazatide. So dalazatide cannot be produced recombinantly. It has a non-recombinant adduct, a non-amino acid adduct, which means it has to be synthesized. That makes it extremely expensive to produce. So we generated, in the last few years, we generated recombinant analogs of dalazatide, and SHK235 has the potency and the selectivity of dalazatide. So now we have something that's recombinant, can be produced much higher speed and also a lot cheaper. And then there was the other problem that everyone told us could not be solved. All those peptides are small peptides. They are folded. They have a very specific structure. So the idea is that they would always have to be injected. You would never be able to give them orally because they would be damaged in the stomach by the acid. They would be damaged by proteases in the small intestine. So oral bioavailability was not a possibility. And we actually believed it. So we decided to go another route. Since we had a recombinant peptide, we decided to instead do tissue-targeted delivery, with the idea that if you're making, for example, a new pancreas for a patient with type 1 diabetes, if it could secrete low amounts of this peptide, it could create a little cloud of immunomodulator and protect itself from the immune system. To make it easy, we thought we would target the liver, because the liver is a huge factory of proteins. It's easy to target things to the liver, so we said, well, we'll use the AAV8 vector that is known to target hepatocytes, and we'll have the liver produce our peptide, and we'll move them to other organs. And that did not work. So it's produced in the liver by very few cells. So what happens is that at first you get a lot of hepatocytes transduced, but over time they're actually eliminated by the immune system, even though they're producing the peptide. And they're producing the peptide at very low amounts. So you're saying, well, why is she showing us that? This is really disappointing and very annoying. Well, the thing is, sometimes you have to fail to move to something bigger. And this failure still taught us a few things. We knew we had a peptide that was recombinant. We knew that there were things that didn't work. So we went back to the drawing board. And going back to the drawing board, we thought again to this oral delivery, right? When you think about patients, a pill is better than an injection, always. So if you're going to do oral and it's not going to be workable to just eat the peptide, it's not going to be workable to put it in an enteric coated capsule, we tried that. So how else could you deliver it if you can make it recombinantly? Well, there are bacteria that a lot of us eat every day. Those are probiotics. So you have them in a lot of food items. And we have a microbiome that contains probiotics. And Rob Britton here at Baylor has the genetic tools to modify probiotics to produce proteins and peptides. So we partnered with him and said, can we make a probiotic that can secrete a venom toxin to treat an autoimmune disease? And it does sound a little bit crazy, but we tried it. So we modified the probiotic, we gavaged rats, and then we took blood from the rats, and we assayed the peptide in the blood. But we didn't want just to see the peptide there. We wanted to see is it functional. Because if it's there but it's unfolded, it doesn't help us. So if we use patch clamp electrophysiology and we patch clamp actually serum samples on single cells, and you do that on a lot of cells, you can see that on the left, the peptide, the bacteria that produces an irrelevant peptide, GAS-A, doesn't block the channel. We don't have a Kv1.3 blocker in the blood of those rats. But if you look on the right, the probiotic LRS235 that produces our peptide, we see a nice block of the Kv1.3 current, showing that we have peptide in the blood of those rats. So the probiotic that we fed to those animals produced the peptide, and the peptide is in the blood of the animals. Is it enough? Is it enough to treat a disease? So we induced a model of rheumatoid arthritis in rats. We immunized rats against their own collagen, and they develop a disease that's very similar to the human disease. In black, you see the vehicle-treated animals develop a fairly severe disease. When we give them in blue the probiotic that does not produce our peptide, if you look very early on, there's a little bit of benefit. Later on, the benefit goes away. So the probiotic itself is not therapeutic. In red, if we gavage the probiotic that's producing the peptide, you can see a complete stop in disease progression in those animals. That was actually quite remarkable because when we inject the peptide, it works, but not quite as well. The first time we did it, we said we messed up, right? So we repeated it, and now it's the third time we get the same results. So we are fairly confident it really works. But I'm showing you numbers there. And you can say, well, what are you measuring? You're measuring a score of disease. Let's get some visual on the paws of those animals. That's going to really give you a better understanding of what's happening there. On the left, is a healthy paw. Those are micro-CT images of the hind limbs of those animals. On the left is a healthy joint. You see healthy joints. The bone is nice, pseudo-colored blue, high density. You see very thin lines where you have the joints. Then you have the vehicle-treated animal with arthritis next to it, and you see that the joints are completely chewed up. You see a lot of red, a lot of bone damage. You see that in all the joints in the paw. Next to it is the animals treated with L-argase, the probiotic that's not producing our peptide. Similar damage, no benefit visible from that probiotic. Next to it is the injected peptide. It's not perfect, but you can see that the joints are much better preserved. Same thing when we gavage them with the probiotic that produces our peptide. The joints are much, much better preserved. So what we have now is we have a selective peptide blocker of the Kv1.3 channel that's selective, that's high affinity, that has efficacy in a small phase 1b trial. So we have high hopes for future of this peptide in clinical trials. We know that tissue-targeted delivery, well, if we want to go back there, it needs a lot more work. But what we now have is that we have a probiotic-based oral delivery that actually works. The oral delivery that we always thought would never work, we now have a way of doing it. And so now the big question we have going in the future is that are we going to be eating yogurts as therapeutics for our diseases? And I thank you.
So thank you very much, Dr. Beaton. And next we have the presentation by Dr. Jeffrey McGee, who gave us a recording of his comments so that we could hear about his work as well.
Well, hello everyone. This is Jeff McGee. Let me start by apologizing for my absence. I'm giving a talk in Europe, probably at this very minute. Let me thank the college for this award, and I'm happy to accept on behalf of the lab, since it is really their hard and smart work that is being recognized. So the lab is trying to understand how brains learn and remember just by interacting with the world. Examples of this range from how we all find our way home from work every night to something more exotic, like how we're able to retain all of the individual episodes of our lives that are so important for our unique sense of self. So let me start on the simple side of that range with an example of a mouse encountering a new environment for the first time. Initially has no idea what the best route through this environment is, but as it explores, adaptive changes occur within its brain. These changes are adaptive in the sense that they improve the behavior of the animal, such that after a few explorations, the brain is able to produce the best route through the environment. Now, some weeks of time may go by here, and a portion of these adaptive changes have been maintained within the mouse's brain, and they allow the brain to produce a pretty good route through the environment. So this is obviously a two-stage process, with the first stage being the formation of these adaptive changes, and I'll call that learning. And the second stage is the retention of some portion of these adaptive changes, and I'll call that memory. Now, we work in a brain area known as the hippocampus. There's been a lot of evidence over the decades that the hippocampus is critically important in memory and learning in animals. And we have recently discovered what we think are the main cellular and circuit level mechanisms of learning and memory in the hippocampus. And that's what I'd like to sketch out for you today. So the hippocampus is home to the world-famous place cell. A place cell is simply a neuron whose activity or firing rate goes up and then down when the animal runs through a particular location or position in the world. We can convert this place field into a heat map where the warmer colors show higher firing rates. So this is the animal's place field with its peak right around this red color. Now, the hippocampus shows a very impressive adaptive change as an animal learns particular important regions in the world. In this case, a mouse is running laps on a track, a sort of NASCAR racetrack. And the experimenter is changing the location of a reward site on every trial or every lap. And you can see that there's essentially nothing for the animal to learn here. The mouse licks everywhere. And under these conditions, if you record from a population of these place cells, there's about 1,000 of them that have been sorted by the location of their place fields, you can see that these place fields or place cells uniformly tile the entire environment. There's essentially one place cell for every location. Now, if on the next day or later that same day, the experimenter fixes the reward at a particular location, you can see that the animal learns this location and changes its licking pattern to only lick that particular position. Now, if we record from the hippocampus again, we can see that there's been a dramatic change in the place cell representation, such that there is now a threefold increase in the density of place cells around the particular location of the reward. And this is truly an adaptive change in the hippocampus, because this group and many others have shown that if they block this overrepresentation or hippocampal activity in general, then this change in the licking behavior, this learning, essentially does not happen. Okay, so we were interested in trying to understand how all of this is happening in the hippocampus. What are some of the mechanisms involved? To look at this, we're going to use electrical recording techniques to record the membrane potential from individual CA1 pyramidal neurons, and then just observe this membrane potential as an animal runs laps on a track. So in the black here is the membrane potential of a given neuron, and in the red is the location of the mouse on one of these NASCAR tracks. You can see with the animal standing initially here, there's some activity in this cell. Here's 5 action potentials. You can see that they are brief, large amplitude voltage spikes that last about a millisecond. But when the animal starts running, you can see that there are no action potentials and really no significant depolarization. And as I start the video, you can see that this is true for the next few trials. There's a little bit of activity while the animal is standing, but as soon as it starts running, it goes essentially silent. No depolarization, no activity again on this trial. And now in this next trial, something dramatic is going to happen. And then after that, there's going to be a fully formed place field at a particular location in this world. You can see there's a slow ramp of depolarization, high frequency firing at a particular location on each of these trials. And we can see then in this last lap that there's a nice place field that's been formed, a slow ramp of depolarization that drives high frequency firing at a particular location. Okay, so if we compare one of these earlier laps in this animal, the membrane potential from one of these earlier laps versus one of the later ones after the place field was formed, we can see that there's this slow ramp of depolarization that's driving high frequency output at the place field. We now know that this slow ramp of depolarization is produced by a new form of synaptic plasticity that increases the weights of the excitatory synaptic inputs that are coming in at this particular location. We named this new plasticity behavioral timescale synaptic plasticity because it's operating over this seconds timescale. And we found that BTSP is induced by the initiation of these very large voltage and long duration voltage signals that we call plateau potentials. So plateau potentials induce BTSP to adjust the weights on a feed-forward input into these cells such that they can now produce place fields. So BTSP produces place fields in the hippocampus. We originally stated the question as what are the mechanisms responsible for this adaptive change? The adaptive change is a population phenomenon. So in order to determine if BTSP is responsible for the adaptive change, we need to record from a population of neurons in CA1. Then we're going to use 2-photon calcium imaging to record from several hundred neurons simultaneously. You can see that in some of these cells, there are bright flashes of increases in fluorescence, and this is occurring at particular locations on the track. If we average up all of these fluorescence recordings from a given neuron, we can see again an increase in activity and then a decrease in the activity around a particular location. So this looks a lot like a standard place field. Now, is BTSP responsible for these adaptive changes? To cut a long story short, Christina was able to reproduce the formation of this overrepresentation that was found in the hippocampal area CA1. So when she fixes a reward site as an animal runs laps on a track, then there's a buildup of over 3 times higher density of place cells around this very salient region of the world. And she was able to block this buildup in the overrepresentation with 2 pharmacological agents that block BTSP. One is just a general synaptic plasticity blocker, and the other is an antagonist that inhibits the plateau potential initiation. She was also able to block the overrepresentation by optogenetically manipulating a circuit element that is responsible for driving plateau potentials and thus inducing BTSP in this circuit. So these 3 pieces of strong evidence tell us that BTSP is capable of underlying the adaptive changes that are responsible for learning in the hippocampus. So BTSP does underlie the adaptive changes. Okay, so that's pretty much where we are with learning then. We found that BTSP driven by plateau potentials is responsible for the adaptive changes that allow the animal to produce the best behavior within a new environment. So we think then that BTSP is one of the most prominent cellular and circuit level mechanisms of learning in the hippocampus. Okay, so that's it for learning. We're also interested in memory formation. Of course, memory is important. In order to look at memory formation in the hippocampus, again, we're going to not just report from this population of place cells for a single day, but we're going to track this population over the course of a week. We can see that there are some place cells that show stable place fields for the week, others that only show stable place fields for about a day. And in fact, we can divide the entire population of place cells up into these 2 groups based on how stable their place fields are, with one of them being about 10 times more stable than the other. So let's look at what's going on here during the week. Here are all the place fields that are formed on the first day. You can see that most of them, about 75%, are these plastic or unstable placed cells. But there's a small fraction, about 25 or 30%, that are quite stable, which means they have consistent place fields for 3 or more days. And then on the next day, something similar happens. We still see these stable place cells, and now there was an addition of another small fraction of stable place cells to this stable representation. And as we track this out over the week, we can see the size of this stable representation is growing at the expense of the plastic group, because the total number of place cells that are present within the population each day, it stays constant. So this looks to us as if we're watching the formation of a memory of this particular task that the animals are undergoing across the week. So again, we see the size of this stable population grow across the week, indicating that the representation itself is stabilizing as the animal continues to learn. We now know that BTSP is in part responsible for this enhanced stabilization during memory formation. Okay, so that's it then. We think that we have recently uncovered the neuronal mechanisms for learning and memory in the hippocampus, and both of these things revolve around this new plasticity, which we've recently discovered, called behavioral timescale synaptic plasticity. Okay, let me then acknowledge the pioneers of this work. Katie, Christina, and Aaron discovered BTSP. Sachin and Guan Chun did all of the work that I showed you on memory. Yiding has a very nice story about BTSP in areas CA3 and other members of the lab currently. Randy keeps the lab running, and we have a long-term collaboration with Sandro at Janelia. Okay, so that's it. Thank you very much for the award. Very much appreciated. Thank you.
So that was a lovely talk, and I'm pleased we were able to have him present that today, albeit a little bit indirectly. And so now I'd like to invite Dr. Maxine Mamonkan to come up and give her his address.
Great. Thank you very much, Dr. Smith, and thank you all for coming today. Today I'll talk a little bit about how we engineer cell therapies against diseases that are driven by malignant pathogenic T cells. And so making new therapies is team science. So I'm going to start with this slide, and I know you are in this room. I'm very grateful to see you here. This work really is a collective effort of people in my lab, present and past, as well as folks in the center, led by Helen Heslop, Malcolm Brenner, and also the clinical team, GMP and GLP folks, and our collaborators. So the therapeutic modality that we are developing is called adoptive cell therapy, which is shown here on the left, where we take patients' own T cells and modify them in the laboratory with viral vectors that encode receptors that essentially teach them how to navigate to the tumor and then kill those tumor cells. And so really there's a balance between what we engineer to put in T cells and how we grow the cells. And this is very important and will become evident later in my talk. Our favorite toy is called the chimeric antigen receptor, which is a chimeric molecule that allows these T cells to directly recognize tumor antigen without having to recognize the conventional MHC and peptides. And so the details of that are not really important for this talk, but what is important is that the clinical outcomes in patients that receive these CAR T cells are phenomenal, particularly those with B-cell leukemia. You can see on the right the survival curves with kids with B-cell acute lymphoblastic leukemia on salvage. Their survival curves look absolutely horrible, but those that receive CD19 CAR T cells have very good outcomes. Now, the problem extending this approach beyond the B-cell paradigm is that the antigens that we can target are not as good as CD19. And in fact, if you look at survival curves in patients with T-cell malignancies, which are really sister malignancies to the B-cell tumors, their survival curves look very bad. And the 2 and 3 year old survivals are hovering around 20%. And the reason for that is really the paucity of good targets. And primary reason why we can't really engineer effective CAR T-cell therapies for that as easy as we could for other diseases, because the antigens that we target on malignant cells are also present on our own therapeutic cells. And that creates this opportunity for them to kill each other before they even go to the patient. So the examples of those antigens are shown on the bottom, CD5 and CD7. You can see that more than 95% of your therapeutic cells express these very targets, and so they will be killed before they even reach the patient. So we thought we would still start somewhere and made a CAR that targets CD5. Again, it's one of the most prominent markers in malignant and normal T cells. And much to our surprise, we found that these CAR T cells actually did not kill each other. And the work that was led by a very talented graduate of mine, Royce, uncovered the mystery of that, which is when we put that CAR in our T cells, that results in very effective downregulation of CD5, a complete degradation of the protein. So essentially, they get rid of the target very quickly and become invisible to each other, and that's what allows them to expand. And once they expand, they're killing tumors really, really well. So they're hiding from themselves, but they're eradicating leukemia in the dish and in mouse models very, very effectively. And so what really this gives us is an opportunity to generate therapeutic cells for patients with the disease without really doing any complex engineering or gene editing or other additional approaches that would be required to overcome the self-targeting of the cells. So that's exactly what we need. And that's what we did. And we developed a clinical trial with Dr. Lakisa Hale and Rain Rouse in patients with T-cell lymphoma and T-cell ALL, adult and pediatric patients respectively. We've recently shown the preliminary results in the first cohort of patients with T-cell lymphoma. We treated 9 patients across 3 dose levels. And we saw responses in 4 out of 9, including 3 complete remissions, which means that the disease becomes undetectable and essentially patients get rid of their cancer. And mind you, these are patients who don't really have any therapeutic options on the table. We're grabbing them from their way to hospice. And what you can see that the CAR T cells expand, and they gradually melt away as the disease melts away as well. And there's an example of that response you can see on the PET CT scan on the right. So a single dose of these CAR T cells is very effective in eliminating malignancy. Now, the small number of patients really makes it very difficult to understand why some respond, why some don't. But one thing that we did notice is that all our responses were achieved with products that were manufactured with a certain way, where we freeze them very soon after cryopreservation. Again, going back to the point that it's very important how you grow the cell, not just what you put in those cells. And so the cells, once we use the shortened manufacturing, we cryopreserve them sooner, they expand in the patient more instead. And they expand and then they persist better, and the quality of that product is a little better. They're enriched for the cells that are highly potent and navigate to the tumor. So that all makes sense to us. What did not make any sense is that the responses in patients with T-cell leukemia were completely different. We didn't really see any notable responses out of 8 patients that we treated. And when we looked at the expansion and persistence of CAR T cells, it was very, very different. They expanded, but then they crashed after week 2. And so as Dr. Beaton mentioned in her talk, you have to fail before you can move forward. And so that really forced us to go back to the drawing board and try to understand what it is that limits the activity of CAR T cells in these patients. One thing that we found out, and this was a project that was led by my technician, Madhu, what we realized is that the CAR is constantly signaling in these T cells. They're always producing the buzz that keeps these T cells on their feet and essentially exhausts them even before they're infused to the patients. And what she also found out is when she adds 2 pharmacologic inhibitors of kinases, dasatinib and ibrutinib, it completely prevents that spontaneous phosphorylation and really mutes all the deleterious CAR signaling. And what it does also, it preserves the highly potent, highly beneficial T cell populations that are otherwise ablated by that continuous buzzing. And we're also seeing much better activity in mice, and the survival of these animals are significantly extended. And so we thought that we could just implement this approach in the GMP. Also, what we do is just we add these 2 inhibitors during CAR T cell manufacturing and not do anything else. And that's exactly what we did. And then we treated several patients with the disease. So this is the original patient cohort that I showed you before, with very transient expansion, pretty much no responses, one very, very short-lived responses. And here are the outcomes of patients that receive the product that was just expanded in the presence of these inhibitors. Very simple change, but now you can see the CAR T cells expand and they last, they persist long-term. And that's exactly what we need in patients with rapidly progressive disease, because now we have an army of cells that continuously attacking the tumor. And as a result, out of 8 patients, we're seeing 6 responders, including 5 with minimal residual disease negative outcomes. And so this really transforms the outcomes in patients, and again reiterates the point that it's not just the engineering, but also how you grow the cells matter. And so we thought that we really have to bring these therapies to as many patients as possible. And as Dr. Smith mentioned, we are opening a large multi-center phase 2 trial to really make it available to everybody who needs it, and hope we'll initiate the trial later this year. The IND was approved 2 weeks ago. So very excited about this, but also another good news for the patients that if we extend this approach when targeting another antigen, CD7, which is also a very good antigen in T-cell malignancies, it also works very well. So as you can see here, another clinical trial that we run with the same investigators show really good evidence of responses on dose level 2. We continue to escalate this trial and really looking forward to the results. Now, so these are excellent news for the patients. Now they have the option for potentially a curative option for their diseases when targeting CD5 and CD7. But can we learn from this experience and develop something for diseases that are driven by pathogenic T cells? So we heard an excellent primer from Dr. Beaton on autoimmunity that is driven by T cells. Also, aberrant activation of T cells can lead to other fatal conditions like rejection of a transplant organ, or the other way around, when the T cells from the transplanted organ recognize host tissues and mount fatal immune attack. And so we thought that we could develop something similar for those fatal conditions. Now, here the targeting has to be much more precise. We cannot just carpet bomb the T cell compartment by targeting a pan T cell antigen. Instead, what we decided to do is target one of the antigens that are very selectively upregulated on activated T cells. And so again, Dr. Beaton showed that KV1.3 is one of the potential targets, and there are additional targets that are not potassium channels, but also mark the activated T cells in their pathogenic state and are not present in the populations that are otherwise harmless. And so the two examples of these antigens are 4-1BB and OX40. As you can see on the right, it goes very transiently up and then it goes down. And so it basically only is expressed in a pathogenic state of the T cells. And 4-1BB stays longer on CD8s, whereas OX40 stays higher on CD4s, and that creates an opportunity for differential targeting of these two populations, depending on what is the driver of your disease. And so we decided to develop these class of receptors we call autoimmune defense receptors that will essentially recognize either of these markers and essentially eliminate the pathogenic population. So in the context of immune rejection, we tested this approach first in vitro. This is a classic model where we mix T cells from one donor with blood cells from another donor, and in this condition, the T cells from that donor are getting rejected within several days. As you can see, the control T cells by day 9 are completely gone. But when they're armed with the ADR that targets 4-1BB, they resist this rejection and actually expand. We can also model that in mice. We also engraft them with recipient T cells and let them be rejected by the allogeneic donor, and control cells are gone, but ADR is back to black. And so you can see that they resist rejection very well. Now, it's great to protect ourselves from immune rejection, but they have to have some therapeutic function. And so we thought, what if we can make a CAR T cells from a healthy donor and then use these CAR T cells in the patient and then utilize ADR to prevent rejection of these therapies and make it last. And so that's exactly what we modeled in this mice, where we essentially implanted a tumor and injected donor CAR T cells that had to resist the immune rejection from the heterologous donor. And you can see that the CAR T cells that did not have ADR, that had very transient expansion and very transient anti-tumor activity. As you can see on the bottom, the tumor burden initially contracted, but then spiraled out of control, killing almost all the mice. But when we overexpressed that ADR in these T cells, the T cells expanded, persisted long-term, and protected the vast majority of animals from the fatal disease. And then finally, we could also model the graft-versus-host disease, which is the other way around, when the T cells in the graft could start recognizing normal, healthy tissue. So we can model that by engrafting normal human peripheral blood molecular cells into mice. And what happens there is that these PBMCs start recognizing mouse tissues and cause graft versus host disease, which eventually kills the animal. And we found that a single injection of T cells that target OX40 through the ADR completely reverses this pathology and protects all the animals from this fatal autoimmunity by preventing the expansion of CD4 cells and ensuring prolonged survival. So to summarize, we can target T cell malignancies with CD5 and CD7 CAR T cells. We've shown this in the clinic. It works really well. It works even better when we inhibit tonic CAR signaling, which turns out to be a very important component, regulating their activity. And we can also utilize a similar approach to target T cell-driven pathologies that are driven by CD8 cells through 4-1BB or by CD4 cells through OX40 ADR T cells. Again, I want to put this slide out there. I haven't held a pipette in years, and that's probably good. And all this work was done by the very talented people that are shown here. And thank you for your attention.
So thank you very much. That was a wonderful presentation. And now we'll turn and invite Dr. Leonid Metalica to come up and to give us a talk about his work.
I'd like to thank again DeBakey Foundation for this great honor. My research mostly evolved around NKT cells. This is a unique subset of lymphocytes sharing property of both T cells and NK cells. And they also have some unique properties. So like T cells, NKT cells have T cell receptor, but NKT cells recognize a unique molecule called CD1D, which present glycolipids instead of peptides, whereas conventional T cells recognize peptides presented by either HLA class 1 or class 2. And NK cells don't have T cell receptor, but they recognize targets based on balance of activating and inhibitory signals. So as I mentioned, NKT cells have some properties of both, as they have markers of T cells and NK cells. And importantly, this population was discovered in the early 90s as a subset of lymphocytes responsible for some anti-tumor properties in mice. And therefore, our initial studies were focused on understanding what the potential role of these cells in humans. And early in my lab, in my independent work after my fellowship, we looked at a number of primary tumors from children with neuroblastoma and analyzed the presence of different immune subsets in these primary tumors. And to our surprise, we found that only NKT cells correlated with outcome. So in this original figure, you can see that patients who had infiltration of NKT cells in the primary tumors had much better long-term survival compared to patients who did not have NKT cells. Of course, this was a correlative finding, and then many years since then, we tried to understand the mechanism, how these cells work, and also try to use natural property of these cells for developing NKT-based cancer monotherapy. In parallel, we also thought to use synthetic receptors, such as chimeric antigen receptor, to enhance natural property of NKT cells. As Max already told you briefly in his presentation, CARs are hybrid molecules consisting of antigen recognition domains of monoclonal antibodies and engineered domains of T cell receptor signaling, providing opportunity to recognize target antigens like antibody and then engage T cell machinery to kill targets and to proliferate and expand. So basically, allowing to use immune effector cells like killing machines. So why NKT cells and not T cells, as originally used as car carriers? First, our lab and many other groups found that NKT cells have unique properties of localizing to tumor sites. That's something that absolutely critical for targeting solid tumors, as your effector cells must localize to the tumor site to have a chance for anti-tumor activity. Second, we found that NKT cells have some unique properties in terms of targeting cells of tumor microenvironment, such as tumor-associated macrophages that promote tumor growth. Also, several groups demonstrated that NKT cells can license dendritic cells to cross-present antigens to CD8 T cells and to engage adaptive immune response, as well as to activate NK cells. So this is really orchestration of multiple aspects of anti-tumor immunity. And finally, CD1D, which is the antigen-presenting molecule for NKT cells, is monomorphic, and therefore it's identical in non-humans. So the NKT cells don't have potential to induce graft versus host disease, and potentially could be used for off-the-shelf immunotherapy. So with this, we developed our first clinical product with NKT cells expanded from patients with neuroblastoma, engineered to express chimeric antigen receptor against clinically validated targets on neuroblastoma cells called GD2. And in this first in-human CAR-NKT cell trial, we tested NKT cells using 3+3 dose escalation format in patients with relapse-resistant neuroblastoma, evaluating safety, anti-tumor toxicity, anti-tumor activity, and correlative evaluation. This study was led by my long-term collaborator, Dr. Hegzi, who is unfortunately not here today. The major challenge for clinical use of these cells was that these are very rare cells in the blood. As you can see here, you can barely see these cells in freshly isolated PBMC. They, on average, represent 0.1% of PBMC. But we developed procedures to expand these cells to clinical scale with very high purity, and at the same time engineer them to express CARs at a similar rate as in T cells. And this is results from all 12 patients tested in our first trial. You can see that on average we achieve 95% purity of the cells and around 50% CAR expression on average. And the absolute number of the cells generated from each patient was around 500 million cells per patient. The whole procedure took around 10 to 11 days. So in terms of safety, in our first evaluation of 12 patients on 3 dose levels, we found very little toxicity. In particular, there was really no grade 3 or higher toxicity related to NKT cell product itself. Most of toxicity was associated with lymphodepleting chemotherapy. Therefore, the product was quite safe. Then we evaluated anti-tumor activity, and as you can see, we could see objective responses in these highly pretreated patients resistant to any other type of therapy. And we could achieve 3 objective responses, including 2 partial responses and 1 complete response. In addition, 2 additional patients observed anti-tumor activity that did not reach the level of objective response, but nevertheless, it was detectable anti-tumor activity based on objective criteria. These are examples of responses. As you can see here, there was a lesion in the sternum, and the lesion decreased dramatically 4 weeks after single injection of current KT cells. And on additional observation 4 weeks later, the lesion almost completely disappeared. So this was still considered partial response because some remaining signal was detectable. And in this patient, we observed complete resolution of multiple metastases, as you can see here, after single infusion of NKT cells. As I mentioned, one of the premises of NKT cells compared to conventional T cells is that they can localize to the tumor site. And we indeed observed that we could find NKT cells in tumor biopsy taken around 2 to 3 weeks post-infusion in all patients in which we could obtain such biopsy. Then, of course, we try to understand what would predict response in these patients. And the most critical observation was that the dose of infusion did not correlate with response. We found that in vivo expansion of NKT cells, that means the dose that achieved in vivo correlated with response. And we found that in a particular marker called CD62L in pre-infusion product highly correlated with the ability of these NKT cells to expand in vivo. Moreover, just expression of CD62L or frequency of CD62L positive current KT cells in the product correlated with response versus no response. Further analyzing this pre-infusion products, we performed single-cell RNA-seq analysis and identified a particular cluster of cells that are highly enriched in CD62L, which is encoded by a gene called CEL. So you can see that this cluster is highly enriched in responders versus non-responders. Of course, CD62L is just one gene in this cluster. We can call it a marker of this cluster. But of course, there are multiple genes here that represent subject of further investigation in our labs. Trying to further understand the role of CD62L and associated cellular subsets, we found that indeed when we expand NKT cells ex vivo, there is a clear enrichment of CD62L positive cells. And in general, these cells called central memory like cells analogous to such subset in T cells. And when we physically sorted NKT cells to CD62L positive and negative subsets and try to re-stimulate them with antigen in vitro, you can see that only CD62L positive cells could undergo multiple rounds of expansion in response to re-stimulation. And when we express a CAR in these cells and treated mice with CD62L positive current KT cells and CD62L negative current KT cells, only CD62L positive cells mediated a durable tumor regressions in mice. And then we performed gene expression analysis in sorted CD612 positive and negative cells, and found there is a number of genes that differentially expressed in these subsets. And in particular, we found that LEF1, known transcription factor that involved in T-cell memory regulation, as well as early stage of NKT cell development in thymus, was particularly overexpressed in CD62L positive subset. So we then tried to understand whether there is a cause-effect relationship between LEF1 and generation of NKT cell memory. And we found that indeed, on the protein level, CD62L positive cells have high expression of CD62L, confirming our RNA-seq results. And then in loss of function experiment, we performed CRISPR-mediated gene knockout for LEF1, and then tried to expand the cells and re-stimulate. We did not see any difference in cell expansion after initial stimulation, but when we tried to re-stimulate cells, only CD62L positive subset could retain, only LEF1 control cells could retain CD62L expression and ability to expand further, suggesting that indeed LEF1 control central memory program in the cells and ability of NKT cells to respond to re-stimulation and long-term persistence. So with this knowledge, we now go back to translational effort and trying to improve our current NKT cell therapy by co-expressing GD2 specific CAR with LEF1 in one construct. And as you can see from this animal experiment, NKT cells co-expressing GD2 CAR and LEF1 mediate much more durable anti-tumor activity in mouse models. So with this, we can now see a potential next generation product that we could test in next clinical trial. So in summary, I hope that I convinced you that we can now generate very pure NKT cells that can express chimeric antigen receptors. They can be manufactured to clinical scale in GMP and used to treat cancer patients. Initial clinical evaluation of NKT cells in patients with neuroblastoma demonstrate that these cells are safe. They can expand in vivo, localized to the tumor site, and mediate anti-tumor activity in neuroblastoma patients. We also found that this anti-tumor activity is associated with the ability of NKT cells to persist and expand in patients, which is associated with expression of CD62L positive central memory-like NKT cells. And we found that this memory cells is driven by activity of transcription factor LEF1, which can be used for further engineering and the development of next generation NKT cell immunotherapy. Now I would like to acknowledge our labs, my labs, as well as Dr. Hegze lab. Our labs now form a new center called Center for Innate Advanced Cell Therapy, formed 2.5 years ago with support from Texas Children's Hospital. And of course, this clinical effort take many collaborators and many support from other groups at CHET, at Texas Children's. In particular, I would like to thank Dr. Heslop, Dr. Brenner, and groups of GMP people and regulatory people who support our GMP and clinical trials. Thank you.
And so our last presentation for this afternoon is by Dr. Bing Zhang, and I'd like to invite him to the podium to tell us about his work.
Thank you, Mary. It's a great honor to receive this prestigious award. Research in my lab focuses on computational cancer proteogenomics and with applications to precision oncology. So first and foremost, I would like to thank my lab members. This award is a testament of the hard work and dedication of the whole group. I'm truly fortunate to have the opportunity to work with this talented and committed group. And as a computational lab, we rely on our collaborators to generate data and to perform experimental validation for our computational predictions. So I'm grateful to our collaborators, both within and outside Baylor, especially the NCI's Clinical Proteome Analysis Consortium, which provide much of the context for our work in cancer proteogenomics. So we know that cancer is a disease of genetic aberrations, and genome-driven precision oncology aims to tailor treatment based on individuals' tumor mutations. So one of the largest clinical effort or clinical trial is the NCI's MATCH trial, which was initiated in 2015 and was completed last year. In this trial, they recruited 6,000 patients, but only 26% of these patients were able to match to any of these sub-studies. And among the 27 completed studies, only 26% of these studies showed positive results. Obviously, this showed that this approach is only partially successful. And this is not surprising if we think about biology, because there are a lot of processes downstream of the genome that may determine cancer phenotype. So rather than just focus on the mutations, cancer proteogenomics tries to integrate mass spectrometry-based proteomics and genomics and transcriptomics in order to gain a more comprehensive understanding of tumors. Hopefully, that will lead to better diagnosis and treatment. I think incorporating proteomics into cancer research is critical, as we already saw. I mean, most of the molecularly targeted cancer drugs, including the immunotherapies, target proteins. So in a typical proteogenomic study, we collect blood samples, primary tumor samples, and adjacent normal samples from at least, let's say, 100 patients, and then perform whole genome sequencing, whole exome sequencing, methylation array, RNA-seq, microRNA-seq, global proteomics, and the PTM proteomics on these samples. And this could generate up to more than 100,000 different features from these assays. And the mission and the interest of my group is try to translate this huge amount of data into useful insights. And we try to achieve this goal using three complementary directions, or performing research in three complementary directions. The first one is to develop comprehensive computational proteogenomic workflows that can handle different types of tasks, from the data processing to quality control to integrative data analysis. So by using Docker, Nextflow, and the AWS online services, we try to make our workflow automated, reproducible, scalable, and cost-effective. And these standard workflows have been applied to the CPTech studies in different cancer types during the past several years, leading to new discoveries in various cancer types. I just use head and neck cancer as an example. Our integrative analysis has defined 3 molecular subtypes. And we also identified dysregulated pathways for each of these subtypes that can be targeted by either FDA-approved or investigational drugs, like CDK4/6 inhibitors, EGFR inhibitors, or checkpoint inhibitors. More importantly, we found that the genomic aberrations in these pathways, for example, the EGFR amplification, cannot accurately predict the pathway activity And this has been used in clinical trials to select patients, right? Instead, we found other like gene expression or the PTM features might better predict the pathway activity. And the study suggests if we use those to select patients, maybe the response rate can be improved. So in addition to perform data analysis, we also try to develop new algorithms, computational algorithms to address unmet need in the field. One example is regarding the isoform analysis. We developed a graph algorithm-based approach that can help us to better quantify protein isoforms in shotgun proteomics, which is a long-standing challenge in the field. And we have also developed a deep learning-based method to improve the phosphopeptide identification from the phosphoproteomic studies. In order to facilitate functional interpretation of the phosphoproteomic findings, for example, we may identify a phosphosite from a CPTEC study, but we don't really know what it does. We have developed transformer-based deep learning models to systematically extract all the sentences describing any phosphosites. And then having these sentences, for example, with this extracted sentence, we can easily connect these identified sites that is altered in tumor to, in this case, to the glycosylation and the ER-associated degradation of PDL1. So this way, we can gain a lot more information from the phosphoproteomic findings. So in addition to analyze the data by ourselves, I think it's also very important to make the data available to the public. CPTEC has generated a lot of data, and the value of this data extends way beyond the initial publications if we can make this data available to the public. So working with our colleagues in the consortium, we have generated a harmonized data set based from all these studies on more than 1,000 patients across 10 cancer types. So these are publicly available and could serve as a good resource, especially for bioinformaticians and computational biologists to reuse the data to either generating hypotheses or test some of their hypotheses. And to make this also available to biologists who cannot easily program, we have also developed the knowledge base, including 40,000 web pages with pre-computed information on genes, proteins, mutations, and phenotypes. So now anyone can just type in a name of a gene and retrieve the information associated with that gene. And more recently, we have integrated the CPTEC data with other public data sets, like the DepMap data through the, with perturbation information in cell lines to identify therapeutic targets. And in this analysis, we were able to identify overexpressed and hyperactivated protein dependencies, synthetic lethal dependencies, and new antigens and tumor-associated antigens as putative new targets. And these predicted proteins and peptide targets and their supporting data are also made available in a user-friendly web portal. And this was just accepted for publication in Cell, which will appear online in August, I think. So our ultimate goal in the future is to, hopefully we can integrate proteogenomics into clinical practice. Think about patients coming to the hospital and going through the standard of care or clinical trial. If we can collect their samples and perform proteogenomic analysis on deep analysis of all these samples, and by integrating with the outcomes, then we will be able to perform data analysis to identify new biomarkers. This will be able to drive new assay development, eventually can be used to generate new diagnostic assays that can do better diagnosis. Meanwhile, we may also be able to identify new targets that can be used to drive new therapy development and, of course, leading to potentially new treatment. Of course, trying to achieve this will require a collaborative effort from different components, including clinical oncology, molecular characterization, data analytics, and biomedical research, all these teams. I think Baylor actually has established strengths, as we can see today, in all these directions. So if we can work together, hopefully, and to integrate this iterative learning platform, hopefully one day we will be able to fully realize the full potential of proteogenomics, leading to, transforming the patient care and improve outcomes. Thank you.
So thank you. I think we have located now the slide or the presentation. I think should come up shortly. Looking to the back.
I'm sorry I can't join you to celebrate the research awards, but congratulations to everyone. Obviously, these are very special awards named after Michael E. DeBakey, and because we like to celebrate the people who do such great work here, this is a reward for their outstanding scientific contributions over the past 3 years. So today's 6 awardees really represent a lot of the quality and the scope of what we do here at Baylor College of Medicine, and I'm really, really proud of the work you all have done. So Bob Atmar, Christine Beaton, Jeffrey McGee, Max Mamankin, Landon Medelitza, and Bing Zhang, congratulations on your outstanding work, and thank you for being faculty members here at Baylor College of Medicine.
Great, and I'm glad we were able to show that today. So at this point, I want to just thank all of our honorees for being here today, for sharing their outstanding science and contributing to such a wonderful afternoon of learning about the accomplishments and really the really outstanding work and the potential it has for impacting future science and the care of our patients. I also want to thank Dr. David Short and Bill Cottle for being here today from the DeBakey Medical Foundation. We're very grateful for your support and so pleased that you could be here with us today. And lastly, I want to invite everyone to continue to celebrate great science this afternoon. We have a reception that'll be out here towards the DeBakey Library. Please join us. I want to thank everyone who was here today and able to participate and listen to all the great science. And I hope we have a wonderful afternoon, and let's adjourn out to the LCAC lobby.